Thank you, Hervé, again. So, my name is Eric TRABLY. So, I'm a researcher at the Laboratoire of Environmental Biotechnology in France, in Narbonne. And I would like to thank the organizing committee and Hervé for having invited me to present our work about electro-fermentation. So I'm not going to talk about micropollutant remediation. Also, some excellent groups in the world are working in electro-remediation. So I will focus today on electro-fermentation. And I have prepared this presentation with my colleague Nicolas BERNET. We're working together on this aspect. So what is electrofermentation is how to combine fermentation and microbial electrochemistry principle to optimize the degradation, but also to optimize the production of biohydrogen and biobased molecules. So the content of my talk is in three parts. First is a bit of the context, few words about the context of the research, and a focus on biohydrogen. Second part is about dark fermentation and MEC, microbial electrolysis cells. So I will give some principles and also some interesting results we got by coupling these two processes. And finally, more fundamental research, I will present more fundamental research on electrofermentation. Is it a step forward in the degradation or the optimization of these processes? So why hydrogen? So we have all heard about the hydrogen energy. So it has some advantages. First, it's highly abundant. It's highly abundant everywhere. And it has a high energy density. It's about twice the energy density of methane. So 122 megajoules per kilogram. So it's very interesting in terms, it's an energetic molecule. And it produces low greenhouse gases emission from its combustion, only water. And it can be an important vector for the energy transition in the near future for us. But it has some main limits. And the main limit is the low volumetric density of these molecules. It's only 10 kilojoules per liter, meaning that it's 1/4 of the methane content in terms of energy per liter. So it creates some problems with the storage and transportation that are really energy consuming. So we have to store hydrogen at 350 or 700 bars and at minus 200 degrees Celsius to store these molecules. So it consumes a lot of energy. And there is a strict regulation for production of storage, of course, because it's a dangerous molecule. And it's mainly present in complex form. That's important because it's not a natural resource. It's really always complex with other molecules. So for that, we need hydrogen processes to extract and recover from different types of resources that I will present. So that's the main point of this energy. It's not a natural energy. But however, it's not only a question of transportation. Hydrogen market already exists in the world. And you can see here the world production of hydrogen is already about 75, 74 million tons per year. In EU, about 10. In France, about 1 million tons per year. And is mainly used in the refinery for the hydrogenation of sulfur compounds when we produce gasoline. So it's also used in the Haber-Bosch process to produce ammonia and to produce some solvents or air and other chemicals. And it's also used in food industry. However, if you look at the transportation here, is almost nothing on the market. And if we consider the hydrogen available for transportation, is also almost nothing. It's about 9% of the hydrogen that can be commercialized and used for transportation. So we need to find new way of production. And is transportation a reality? So I provided few examples. So there are a lot of examples now in the world, but few examples around here. So if you go to Paris, you can take a Hype taxi. So there is a float of around 700 vehicles, light vehicles in Paris, all running. So they are blue like this, running with hydrogen. So hydrogen station is about 700 bars, and filling time is quite short. It's only 5 minutes, and autonomy is as high as the best electric car, 600 kilometers. So for some lines, also, when you don't have electricity, like here, so there are a few examples in Germany, in France, and Europe, you have trains. I think that's, I mean, they plan to be the future, EV vehicles, trains, trucks, rather than light vehicles. And they plan also, you may have heard about that, the Airbus planes by 2035 to use hydrogen as kerosene. So we need a huge amount of energy linked to hydrogen. And the Hydrogen Council, which is a lot of experts in the domain, they calculated the amount of hydrogen expected by 2050 regarding the global energy demand. And if you look at the demand, it will increase, so we are around here, by a factor of 8 in total by 2050. So it will be a huge demand, but it's not only for transportation, as I mentioned, but it's also for power generation and buffering. When you have an overflow of electricity, we can buffer with hydrogen, and of course electrolysis here. It's also a huge amount of hydrogen for the decarbonization of the industry is very important. So for the industrial energy and building heat and power for everyone. So we need new sources of hydrogen. And as I explained, we need to extract and convert some natural resources. And here, this is the current production of hydrogen in Europe. And if you look at the resources, is natural gas, mainly oil and coal. So at 90% is produced from fossil fuels, and only 10% from electrolyzers, from water. And also, electricity can be produced from fossil fuels also. So the environmental impact can be quite important here. So interestingly, we have divided the production of hydrogen in two types of technologies. One is from the resources of energy. So one is the fossil fuel energy here with hydrocarbon reforming. So is what we are talking about currently. The hydrogen production, it comes from this way. And now if we are interested in renewable sources, meaning that power, electrical power, windmill or solar panels, we can produce by electrolysis and water splitting the hydrogen. So it can be a green hydrogen. And even greener, if we are interested in the biomass, and including waste, the subject of the talk today, waste and effluents, we can produce by two sets of technologies. One are thermochemical processes, gasification or thermogasification, and biological processes. And within the biological processes, we have four sets of technologies that I will detail here. So one is a water biophotolysis. It's well-known technology where microalgae are able to do water splitting by using light. So the advantage is the direct production of hydrogen, but the limits are the light. And also we need to control the medium composition. So it's not really adapted to treat effluent from the industry or wastewater. The second type of technology is really linked, is a photofermentation. So it's carried by purple non-sulfurous bacteria. In nitrogen starvation, they produce hydrogen, so it can be used with effluent but it requires light and the control of the nitrogen content in the effluent. So today, if we focus on effluent and we do not really control the composition of this effluent, there are two types of technologies that can fit with this production. Is dark fermentation, which is a classical fermentation where we use fermentative bacteria at low pH, and it suits with a waste effluent degradation and conversion. However, as you will see, conversion yields are limited, quite limited. And the last technology, or the microbial electrolysis cells, MECs. Here, the main support catalyzers are electroactive bacteria that are able to convert the organic matter to electrons, and then we do, and I will explain, water splitting in this process. So effluents, or kind of, effluents can be used. It's quite intensive. However, we need to add a little bit of power in the system that the reaction can occur. So this is the main context. So in terms of life cycle analysis, dark fermentation or biological processes in general have the lowest impact in almost all criteria than other technologies. So it's quite interesting in terms of environmental impact. However, you will see maybe the intensity or the conversion efficiency are not that high. So let's go in detail in the dark fermentation and microbial electrolysis cells. So principles and challenges. What are the main aspects on these technologies? So first, a few years ago, we did a literature survey of all articles and patents dealing with these two types of technologies. So here you have a summary of the articles and patents. So we started with 2,600 articles. So it is quite more complex than only 4 words like this, anaerobic fermentation in mixed culture. So we used a quite complex research. However, it was around anaerobic fermentation in mixed culture, and we removed all the papers dealing with methane only, pure culture, and also review paper. We wanted to focus on technical papers. And here are the results in terms of distribution of the papers. More than 75% of the papers, of course, were dealing with dark fermentation. 10% on dark fermentation and anaerobic digestion, because it's part of the anaerobic digestion. And only few amount of papers, around 5% on MEC only. We were quite surprised. And 1% on coupling dark fermentation and MEC, what I'm going to present today. And also about 10% with photofermentation and coupling these two processes. That could be interesting as well. And if we compare this with the patent, it's quite interesting here. The dark fermentation and dark fermentation coupled to anaerobic digestion, we have less patents. So the dynamics were more towards MEC and coupling dark fermentation and MEC. So there is a strong dynamics on developing this kind of process to improve the hydrogen efficiency. So now in details, more in details, we made some calculations. So what is the dark fermentation? Representing 75% of the articles in this domain. So it's part of the anaerobic digestion pathway. So from carbohydrates, we have 4 steps: hydrolysis, acidogenesis, acetogenesis, and finally methanogenesis. So if we stop the degradation process to this step, we can accumulate VFA, acetate, butyrate, and also hydrogen. So we need to stop here the methanogenesis. So hydrogen is found in every natural ecosystem, and mainly because hydrogen is an electron vector, and microorganisms like to use this electron vector for their own respiration. And finally, in dark fermentation reactor, we stop the methanogenesis. It's quite easy because we practice a heat shock treatment, so it's not the main factor. Heat shock treatment, we stop, we kill the methanogens, and then playing with the HRT, low HRT, only a few hours to 1 day, and low pH, around 5.5, methanogenesis cannot occur. However, as you will see, we have other type of interactions. It's just because mainly the main producers of hydrogen are Clostridium species, this type of species. So they stop here. They are acidogenic bacteria able to accumulate hydrogen as a release of a surplus of electrons in their metabolism and through the acetate and butyrate pathway. And often if we have an overflow of this of the electrons inside the microorganism, they shift the metabolic pathway to lactate or to the ABE pathways. So it's called solventogenesis. So as soon as we accumulate VFA, too much VFA or hydrogen, they switch here to another type of non-producing pathway. So these are the metabolic limitations. On top of that, in mixed culture, so when we treat effluent and waste, we have also ecological limitations, and mainly the most important are lactic acid bacteria. They outcompete quite easily Clostridium species. They are growing under the same condition, and they are using carbohydrates to produce lactate. And all these arrows are pathways that can use hydrogen as a source of electrons for their own pathway. So they produce propionate, we can produce propionate, caproate, and so on. And it can be a source also of electrons to produce new type of molecules. Sorry. So just remember that it's not only metabolic but also ecological limitations that we have to play with. So in 2000, around 2000, a paper they reached the high yield fermentation here, high yield dark fermentation using enzymes. So 12 moles, thank you, Hervé. 12 moles of hydrogen per mole of glucose here. So this is the maximum we can reach. But in reality, what happens is we have the acetate pathway, we produce 4 moles of hydrogen, and acetate, butyrate pathway, 2 moles of hydrogen and butyrate, and a mixed pathway, acetate ethanol pathway, 2 moles of hydrogen. And in mixed culture, the maximum, we assume the maximum we can reach is about 2.5 moles of hydrogen per mole of glucose. And this is important because you can see here you have different VFA that are concomitantly produced with hydrogen, and this represents around 20% of the COD per gram of COD of glucose only. So 80% of the energy remains in the VFA. So if we look back to the literature, what can affect the metabolic network or the metabolic population mostly is a substrate. So in literature, we found that the substrate is very important with, of course, higher production with simple sugars, and then it can decrease regarding the complexity of the organic waste. So simple sugar, industrial effluents. But overall, we are around 10% only, if we consider all the papers, 10% of the COD converted to hydrogen. Of course, we have outliers here, but the maximum is 33%. But one good point, if we look at the operating parameters in all papers, you can see here is quite a robust process, because dark fermentation is about 10% all along, whatever the condition, mesophilic, thermophilic, whatever the volume of the reactor and the mode of operation, batch, fed batch, or continuous reactor. So that means, in one way, it's limited conversion yield, but also it's quite a robust process that can be used for any type or any operating conditions as long as it's suitable with hydrogen producers. So in summary, for the dark fermentation process, you have organic matter. We produce hydrogen and organic acids, some alcohols, some biomass, and undegraded organic matter. The maximum theoretical yield, which is 4 moles of hydrogen per mole of glucose, represents only 33% of conversion. And in the literature, the average yield we found was 10%. So here we have quite an increase. We should reach this 33%. However, the energetical conversion efficiency is very high because we don't provide a lot of energy. I mean, it's just a fermentation. The delta G is very low. It's minus 200 mainly. So bacteria are able to convert the organic matter by themselves. And the main advantages are here. So high organic loads, well-proved technology for a large operating conditions I showed you. So and is well adapted, therefore, for waste and wastewater treatment. However, there are some metabolic limitations and a limited yield of hydrogen. So we need to couple this to other processes. So the most obvious process is anaerobic digestion that I won't present today. I don't have time. But at short term, it's really to couple these two. But what we focused on is how to convert the VFA, so 80% of the remaining COD, to hydrogen. So for this, one technology, interesting technology, representing 5% of the publications, articles, is MEC, Microbial Electrolysis Cells. You will have a talk this afternoon in details of this technology. And here, organic acid and alcohols can be efficiently converted to CO2 by respiration. And we use the capacity of electroactive bacteria to transfer the electrons from their respiration to the electrode here. So the electroactive bacteria are able to transfer electrons to a material. And then by applying here a voltage, we are able to reduce the protons here at the cathode to produce hydrogen. So there are some advantages. So how is it working? How is it working? Is bacteria are able to convert organic matter here. And you can have mediators here, or non-electroactive bacteria as well, but mediators that can be reduced or oxidized on the surface of the electron. You can have a direct transfer through cytochromes, which are small proteins, or through cytochromes and nanowire. So all these pathways are well known, depending on the type of electroactive bacteria. It's important for the electrofermentation part. And MEC, the benefit is if you start from water, you split water to produce hydrogen, is quite a huge amount of difference of potential. But if you start from organic compound like acetate, the difference is lower. So if you compare the potential difference between MEC here and a water electrolyzer, you can see that the difference of potential we should apply is around 0.2 volts compared to 1.2, 2 volts in the electrolyzer. So it's 10 times lower, more or less, in MEC, because the electrons comes from the organic acids. So, and what I wanted to highlight also is the maximum theoretical yield is about 100%. All organic acids can be converted to electrons. And the average yield in publication is 50%. So it's a high hydrogen production yield, high energetical conversion yield, and also hydrogen is almost purified here at the cathode. So in a pilot, we reach about 99.5% of pure hydrogen at the cathodic compartment. However, the limits are high material cost and low organic load in the system. So I wanted to show you some results, practical results on coupling dark fermentation and MEC, because it's quite obvious that we can use the organic acid in this system. So for that, we try to apply this concept on agro-industrial wastewaters, a set of agro-industrial wastewaters, by coupling dark fermentation and MEC and comparing the efficiency of the systems. So in the first step, in dark fermentation first, it really depends on the type of substrate. And here you can see the hydrogen yield compared to the type of substrate. So from cheese whey, fruit juice wastewater treatment plant, fruit processing wastewater, sugar wastewater here, and some other agro-industrial. So depending on the type of wastewater, we have reached different hydrogen yield. In the second step, we produced hydrogen. I will show you the results. But what is important, we made an energy balance in this system. And you can see here that's the energy applied. And for all effluent, we recovered in brown here up to 700% of energy recovered in form of hydrogen than the energy we applied. So it's quite efficient process here at lab scale. And the COD removal in this system ranged between 40 to 60%. So it's also efficient in terms of COD or VFA removal coming from fermentation. So here's the total results of the system. So hydrogen yield in fermentation, and you can compare the hydrogen yield found in MEC. And there is a factor between 6 to 16 times more hydrogen in the second compartment. Is more efficient in terms of yield. And the overall yield, so here is, excuse me, a volume. But if you convert this volume to moles of hydrogen per gram or per mole of glucose, we reach around 10 moles of hydrogen per mole of glucose, which is quite close to the maximum limit of 12. So it was really efficient on this kind of effluent, and we reach a 70% COD removal in average by coupling these two processes. So it is quite interesting in terms of efficiency. However, we can still think that maybe dark fermentation is not efficient enough to produce hydrogen. So I wanted just to show you a new approach we are developing is focusing on the fermentation to produce a molecule, so a different set of VFA. So in this project, a Biotechnologie project, we try to convert the feedstock, different feedstock waste, to a set of jet fuels with a calibrated type of jet fuel. For that, the first step here we are focusing on is the coupling dark fermentation and MEC. And then carboxylic acid, propionic acid, and hydrogen will be converted in the second step with some input from thermogasification here. In the second step, will be converted to butanediol and acetylene and so on to jet fuel. And I wanted to focus on this aspect because fermentation here, we won't produce any hydrogen. It won't be the purpose, but it will be to focus on metabolite production. And then a new aspect of the MEC, it's also to polish, to degrade non-usable or unwanted metabolites to produce hydrogen. And like this, we can have an effluent enriched in what we want as a metabolite and also hydrogen that can be further converted. So it's also another aspect of the MEC that can be used as a polishing process. That would be interesting. So now we can have this question. And if we put MEC and dark fermentation together, because operating conditions are quite similar, and finally we can process the fermentation and the MEC process in the same compartment. And so what we developed in the last years is an electro-fermentation approach. So is it a step forward than a simple MEC or microbial electrochemical conversion? So electrofermentation, what is this? It consists in operating the fermentation of an energy-rich substrate, such as carbohydrate or alcohol, in presence here of an electrode or electroactive bacteria to provide an external or supplementary electron source or electron sink. And like this, the thermodynamic limitation can be overcome in some aspect and to control the metabolic pathways, what I'm going to show you. So there is a difference between electrosynthesis and electrofermentation. Electrosynthesis is when electrons, in terms of amount of electrons, are directly converted to a product. So we have a certain amount of electrons, it goes directly to a product. This is what we call electrosynthesis. Electrofermentation is not the same purpose. It's only to control here the metabolic pathway of reacting bacteria by changing the NAD/NADH ratio in the cells. And like this, metabolic pathway can be, could be controlled by playing with the ORP, the redox potential. So the idea was to trigger some metabolic pathway in this aspect, or if it's not directly with electroactive bacteria, it can be a support here of the electroactive bacteria as electron sink or source of the fermentative bacteria. So for electrofermentation, I'm sorry, I think I missed a slide here. Sorry. So electrofermentation, the reactions are not supported not only by the electrical current, but mainly by the presence of an energy-rich substrate. So electrical current is not the product of the interacts or the main energy source. And as I said, electric current is just a trigger, allowing a fermentation that normally is not thermodynamically not feasible. And the main difference between electrofermentation here and microbial electrosynthesis is it doesn't require high current density to occur. So low energy, and it's just a trigger of the metabolic pathway. So now, what are the examples? So we did some electrofermentation of glucose in maize culture in presence of electrodes, so polarized electrodes, in batch test. So here you can see the metabolic distribution. So in the control is an open circuit, no electrodes, it was a classical batch. So you can see here a lot of lactate in this system with this kind of consortium, ethanol and acetate and a little bit of hydrogen on top of that. And we applied different type of potential. So minus 0.9 volt up to plus 0.9 volt on the working electrode. And what happened here, you can see a shift, a clear shift of the metabolic distribution from lactate to ethanol, in this case, or butyrate and acetate. But we didn't find any relation between the voltage here and the shift, the metabolic shift so far. So what we did a microbial characterization of this system, and we found that in the control we had Enterobacteriaceae in red and Streptococcaceae, so lactic acid bacteria, in blue. By applying a difference of potential with a low current we observed here, we showed a clear shift towards Enterobacteriaceae, which are also hydrogen producers, not as efficient as Clostridium, but also hydrogen producers. So a clear shift of Bacteriaceae in all conditions. And then other type Clostridiaceae here and some remaining Streptococcaceae. So we had a mixture of metabolite pathway due to the selection of and clear selection of Enterobacteriaceae in this system at the end of the batch fermentation. So here with the PCA analysis, you can see that it's clear and is well known also. We confirm this result that Streptococcus casei as lactic acid bacteria correlated with the lactic acid accumulation. Clostridiaceae with hydrogen butyrate and other Enterobacteriaceae with acetate ethanol succinate accumulation, which is quite obvious. But what was important in this system is that we selected the bacteria specifically by applying a very small difference of potential but with very small electrical density, current density. So hypothesis we made was about the microbial diversity shift. So the presence of electrode caused clearly the microbial diversity shift. But the metabolic shift linked to the microbial was linked to the presence of the electrode in the bulk or to the presence of electroactive bacteria. That was the question. That is the question. So what we did then is we only use electroactive bacteria. Geobacter sulfureducens is the most known electroactive bacteria in batch test with glucose and acetate at low pH and temperature. So like this, fermentative bacteria can grow. And what we found is on the right, you have the metabolic distribution. So with the mixed culture, we have this distribution, mostly in this case, butanediol and butyrate. And when we added geobacter, we found a shift again by just adding, no electrode, just adding geobacter sulfureducens, a clear shift towards butyrate here in light green, light green here, butyrate, less ethanol and butanediol, butyrate fermentation pathway. And if we look at the microbial community, I'm sorry I didn't put the names here, but we turn the microbial community from red and blue species to more blue species related to Clostridiaceae species. And Geobacter sulfureducens was only a minority abundant here species in brown. So meaning that it's not only the electrode, but in our opinion, one hypothesis is like electron transfer between these electroactive bacteria and other bacteria in the system can trigger the metabolic pathway of the fermentative bacteria. So it's what I'm going to show you, is the presence only of Geobacter sulfureducens cause a shift in fermentation product and microbial population. So hypothesis again is there any interspecies electron transfer between the bacteria, and can it occur in mixed culture fermentation? So we looked at old results with a new eye. So these results were performed in chemostat with glucose and 8 different inoculum. And after a steady state, so it was really chemostat, glucose was sterilized. With chemostat, we got 8 different steady state ecosystems. So unsurprisingly, on the left, you have the results. So with 8 different inocula, we found 8 different performances because it's really linked to the microbial community. So on red, the hydrogen yield from 1 to 2.5, and blue the productivity from 9 to 5 here for the lowest. However, at that time, we performed the DNA fingerprinting, and you can see here some DNA fingerprints with one abundant bacteria, always the same, so that was surprising, and subdominant bacteria here. So the hypothesis is what the minority species was very important to control the metabolic pathway of the fermentative bacteria. The fermentative bacteria was Clostridium pasteuriamonum, almost in all cases, and minority species were E. coli, Enterococcus, or Lactobacillus. So the hypothesis at that time was a key role of this minority bacteria, and you will see that it can be linked to interspecies electron transfer as well. So what we did also, we tried to reintroduce the species directly in the ecosystem, in the microbial community. So we tried to reintroduce the species we identified previously in the microbial community. And what we found is different behaviors. The first one is no significant effect. We add the species, nothing happens different. So it was the case of Enterococcus species. In another type of behavior, it's a classical microbial competition. In this case, we added again Clostridium pastorianum in the system, so the fermentative bacteria. And what happened is between EcoA and Clostridium pastorianum, a competition between these two, and with average hydrogen yield between these two ecosystems. The third behavior, a bit more interesting, is a synergetic effect where when we added E. coli here, you can see the hydrogen yield was increased by 2.5%. But at that time, some other groups are working on adding anaerobic facultative microbes to degrade the traces of oxygen in the medium. Like this Clostridium can work more easily towards hydrogen production. So in that time, it can be due to oxygen depletion at start of the growth. But what we didn't explain was this effect with Ralstonia eutropha, now his name is Cupriavidus necator, which is a strict aerobic bacteria. So we tried anyway these bacteria, and you can see here the ecosystem the performances were multiplied by 3.5, so quite a lot. And if we look at the metabolites, we observe again a shift between the butyrate lactate fermentation towards acetate and butyrate, which is clearly hydrogen producing pathways. So the presence of a non-growing aerobic bacteria in this system was triggering the metabolic pathway. So finally, with a group here, they focus because the problem with oxygen-consuming bacteria that we can have traces of oxygen. So they focus more on sulfate, that's something we can control, sulfate-reducing bacteria. So they did a co-culture of Clostridium acetobutylicum, so with the BIP Marseille here. Clostridium acetobutylicum fermentative bacteria with the sulfate-reducing bacteria. Interestingly, in that case, with no sulfate, so not able to grow, so alone in pure culture, Clostridium acetobutylicum was able to grow, and Desulfovibrio vulgaris not able, of course. But in co-culture, you can see here, Clostridium acetobutylicum and Desulfovibrio with no sulfate again were able to grow together. I should mention here that if you look at so here it's expressed in genome copies. If you look at the genome copies, we have a bit lower genome copies in co-culture than in pure culture for Clostridium acetobutylicum. It's quite important because I will present some hypotheses on parasitism of this kind of bacteria. And Desulfovibrio vulgaris is growing well here. The effect on the metabolism, so we did some metatranscriptomic analysis, and what we found is clearly a shift in Clostridium pastorianum from lactate pathway here. In co-culture, it was multiplied by 8 times higher acetate butyrate, and then hydrogen was multiplied, so amount of hydrogen by 2.5. So it's quite consistent with what we observed previously. And finally, with a picture, we found that it was a close interaction, direct interspecies electron transfer here between Clostridium in green and here in red, Desulfovibrio vulgaris. So in co-culture, you can see that some of them were totally yellow, meaning that they are exchanging some material in their system. So without an electron acceptor in the system, Desulfovibrio vulgaris was able, the hypothesis was this microorganism was able to provide electrons or to provide electrons to Clostridium acetobutylicum. So we performed, or we did this hypothesis, and then we did some theoretical approach on this interaction. And one question is it electronic parasitism of Desulfovibrio or Ralstonia eutropha or Cupriovidis necator, or other electroactive bacteria, that when they are not in favorable conditions for growing, they are able to use another microorganisms as electron acceptor or electron donor. So here you have a conceptual graph of what we think it can occur in this system. The electroactive bacteria normally are able to provide electrons to the anode, to minerals, to a material to grow. And in some aspects, if they don't have any electron acceptor, they can use electron accepting microbes here. So that was the hypothesis, and especially fermentative bacteria. So it can be used as a trigger also of a fermentative bacteria. So to go a little bit deeper in this aspect, what we did is we focus on one specific fermentative bacteria, again, Clostridium pastorianum, and one well-known electroactive bacteria, Geobacter sulfureducens. So what we did in that case, it was more to focus on a simpler metabolic pathway, the glycerol fermentation, because there is a direct link here between glycerol and propanediol. So it can be directly converted to propanediol by reduction. And Geobacter sulfureducens, so the hypothesis was to use acetate in the medium, but with no electron acceptor in the system. So what could do Geobacter sulfureducens is only provide the electrons to Clostridium pastorianum, only, in absence of electrode. So in pure culture, Clostridium pastorianum is able to convert glycerol in different set of different types of metabolites in mixture, mainly 1,3-propanediol. And in presence of Geobacter sulfureducens, acetate is oxidized and electrons are provided to Clostridium pastorianum. And if we look at the carbon distribution, so carbon dioxide is not provided here. With pure culture, in blue, you have the propanediol, and in orange, the butanol accumulation. And in co-culture, what happened, again, triggered the metabolic pathway towards propanediol, meaning that we have a clear and obvious observation that electrons coming from Geobacter were used by Clostridium pastorianum to another pathway here. However, it's the same as Clostridium pastorianum and Desulfovibrio vulgaris. The biomass, the total biomass, decreases, meaning that it's kind of electronic parasitism during this process. But Geobacter sulfureducens was able to grow with acetate only as electron donor in this system. So it's a way for them to survive in this system. And finally, to finish, what we did is a metatranscriptomic analysis. We tried to understand what were the mechanisms behind this transfer of electrons. Was it direct transfer of electrons or through cytochromes or with mediators? And what we found here is with a metatranscriptomic analysis, we found two potential pathways. So one through a mediator, so some kinds of cobamide, that can transfer from Geobacter sulfureducens to Clostridium pastorianum, and another way through cytochromes here that were overexpressed in co-culture, and the electrons can go through this pathway also. So we are still not sure of the pathway because these two pathways were improved during the co-culture. So the main question now we have is at which extent this interspecies electron transfer is generic? Can it occur with all types of fermentative or electroactive bacteria? And the effect of electrofermentation we found, is it a direct effect on the microbial population, or is just a selection of electroactive bacteria and then this type of reaction can occur as well? So there is still a lot of questions in this aspect, but I showed you that the fermentation pathway in co-culture or in presence of electrode can be very important in terms of control or even improving some metabolic pathway quite efficiently. So the main conclusion of my talk today is about the environmental biorefinery for biohydrogen and also fermentation. So first, coupling dark fermentation and microbial electrolysis, we think is a promising way to produce hydrogen in this case, or to polish fermentation effluent. So up to 10 moles of hydrogen per mole of glucose. I should say that at lab scale is quite efficient, and now the most important is the upscaling process, and we have to focus on this aspect. All groups on the upscaling and the limitation of upscaling in this process. And electrofermentation, in our opinion, is one way to better control the population selection and also the fermentation process when we want to optimize a fermentation pathway. And there is still a large field of investigation of interspecies electron transfer. So there are many groups working also in electromicrobiology. In co-culture, we assume that it has a great potential to overcome some thermodynamics limitation and then better control the metabolic pathway. So thank you very much for your attention, and thank you for all my colleagues in the lab working in this, on this field. Thank you.